Welcome to The Explainer. So, today we are looking at a fascinating biological mystery. When people talk about ayahuasca, they almost always focus on the visionary molecule, DMT. But actually, ayahuasca has two halves, and today we're exploring the half that nobody drinks for the visions. We're diving deep into the neuroscience of the Harmala Alkaloids. These are the unsung chemical stagehands that make the entire ayahuasca experience literally possible. But as we're going to see, their story goes way, way beyond ancient traditions, leading us straight into the cutting edge of modern regenerative medicine.
Okay, let's dive into this. Here's our roadmap for today's explainer. 1. The unsung stagehands. 2. Hacking the gut. 3. Unlocking oral DMT. 4. A second life. 5. The real danger. And finally, number 6. The honest bottom line.
Part 1. The unsung stagehands.
It all starts with what feels like a complete geographic impossibility. On one side of the globe, you've got Peganum harmala, which is commonly known as Syrian rue. It's this scrubby, invasive weed found out in the deserts of the Middle East and Central Asia. Now, on the exact opposite side of the world, thousands of miles away in the lush Amazon rainforest, we have Banisteriopsis caapi, the famous ayahuasca vine. These two plants are completely unrelated. Yet through this absolutely wild trick of independent evolution, they both loaded themselves up with the exact same family of beta-carboline alkaloids, harmine, harmaline, and tetrahydroharmine, or THH for short.
But here is the catch about these incredible molecules. On their own, they are essentially sub-hallucinogenic. They have a very weak affinity for the 5-HT2A receptor, which, if you didn't know, is the biological target that defines classic psychedelics like LSD or psilocybin. So if you take these alkaloids by themselves, you're not going to experience some visionary perceptual reorganization. Instead, at higher doses, you'll just get heavy sedation, a dreamy state, intense nausea, and a very specific physical tremor. In fact, harmaline is such a reliable inducer of tremors that scientists literally use it as a standard animal model for essential tremor in movement disorder research. So yeah, they are the pharmacological stagehands, definitely not the star of the show.
Part 2. Hacking the gut. MAO-A. The real magic of the Harmala alkaloids is entirely enzymatic. These alkaloids are REMAs. That stands for reversible inhibitors of monoamine oxidase A. Think of MAO-A as your gut's chemical security system. It's an enzyme lining your digestive tract and liver, and its sole job is to break down monoamines like serotonin, dopamine, and various tryptamines, while harmine and harmaline are extraordinarily potent inhibitors of this exact enzyme. They temporarily occupy the enzyme's active site, effectively shutting down that security system. And because they are reversible inhibitors, this blockade isn't permanent, right? It fades as the drug clears your system, which usually leaves you with an operational window of about 2 hours. Part 3. Unlocking oral DMT. So under normal circumstances, swallowing pure DMT does absolutely nothing. That gut MAO-A enzyme we just talked about acts like a giant biological wood chipper, completely destroying the DMT through first-pass metabolism before it can ever reach your bloodstream. But introduce the Harmala alkaloids from the caapi vine, and they temporarily jam the gears of that wood chipper. Suddenly, the swallowed DMT actually survives the digestive tract, enters the bloodstream, crosses right into the brain, and boom, an otherwise inert oral dose becomes a profound 5-hour visionary journey. So the crucial point is that this mechanism is purely chemical, and modern science has completely proven it. If you compare the complex traditional ayahuasca brew, which is made of the vine and a DMT-containing plant, to a stripped-down laboratory version called pharmacology, which uses just pure isolated harmine and synthetic oral DMT, the end result is the exact same enzymatic synergy and activation. That laboratory version proves the beta-carboline's contribution is strictly about blocking the enzyme, though in the traditional brew, that third alkaloid, THH, does add a subtle, gentle serotonin reuptake inhibiting effect that kind of shapes the emotional tone of the whole experience.
Part 4. A second life, DYRK1A.
Entirely separate from their role in psychedelic traditions, Harmala alkaloids have a mind-blowing second life. Harmine has recently been identified as an exceptionally potent inhibitor of an enzyme called DYRK1A. Now, this is a kinase that is absolutely central to how our cells control their life cycles, particularly when it comes to neurodevelopment and neuronal function. Harmine inhibits this kinase with an incredibly strong potency, an IC50 of roughly 80 nanomolar. Basically, that means it takes a remarkably tiny amount of it to have a massive biological effect. This singular property has made harmine one of the most exciting lead molecules in some very unexpected fields of medical research.
And this brilliantly illustrates exactly why researchers are so excited. Harmine is literally the first drug capable of making adult human pancreatic beta cells replicate.
A team at Mount Sinai discovered that by inhibiting DYRK1A, harmine can actually force the insulin-producing beta cells, which are the exact cells lost in diabetes, to regenerate. For a really long time, we thought these adult human cells were fundamentally non-regenerable. No way to fix them. But harmine proved otherwise. Furthermore, when researchers synergized harmine with GLP-1 receptor agonists, like those super popular semaglutide weight loss drugs you've been hearing about, they saw human beta cell mass increase several-fold. It is a real game changer.
But wait, it doesn't stop at diabetes. 57%. In laboratory studies, harmine drove a massive 57% rise in proliferating human neural progenitor cells. It stimulates adult neurogenesis, which is the birth of brand new neurons. Beyond that, in Alzheimer's disease models, because DYRK1A phosphorylates the tau protein at disease-relevant sites, harmine actually reduces tau hyperphosphorylation. It also raises levels of BDNF, which is a crucial brain growth factor, showing distinct neuroprotective and antidepressant signals.
Okay, actually, scratch that. Let's pump the brakes just a bit. We have to stay grounded. There is a crucial caveat here. All of these exciting, groundbreaking discoveries regarding diabetes, neurogenesis, and Alzheimer's are heavily preclinical. We are talking about mechanisms observed in petri dishes and animal models, not human therapies you can go get from your doctor today. Also, the regenerative medical research being done right now relies heavily on highly optimized, engineered, kinase-selective derivatives of harmine, not the raw plant alkaloids you'd extract from a vine or a seed.
Part 5. The real danger. Interactions.
Listen, the absolute most vital thing to understand today is that the exact same MAO-A inhibition that makes these alkaloids useful also makes them incredibly dangerous. This is highly, highly interaction-prone chemistry. If you combine these alkaloids with everyday serotonergic medications like SSRIs, SNRIs, triptans, or tramadol, you risk lethal serotonin syndrome, where your brain's serotonin is driven to toxic, potentially fatal levels. There's also the risk of a hypertensive crisis if you consume tyramine-rich foods like aged cheese or cured meats, because that blocked gut enzyme allows tyramine to flood your system and dangerously spike your blood pressure. And finally, the raw Syrian root plant itself is genuinely toxic in a high overdose, leading to severe organ injury. The danger here is very, very real.
Complicating all these dangers is a massive legal mismatch. In the United States, as of 2026, the Harmala alkaloids themselves, as well as the Syrian root plant, are generally unscheduled. Anyone can easily just buy the seeds online. However, DMT, the molecule that these alkaloids activate, is strictly a Schedule I controlled substance. This creates a really strange landscape where the powerful, highly dangerous enzyme inhibitor is essentially treated as a common weed, while the visionary compound it enables is heavily criminalized.
Part 6. The honest bottom line.
Before we summarize, I must be completely clear. This explainer is strictly for educational purposes and absolutely does not constitute medical advice. Because of the profound drug interactions we just discussed, anyone taking psychiatric or other serotonergic medications must consider Harmala alkaloids completely contraindicated. A suspected serotonin syndrome is a medical emergency. Full stop. Our goal here is purely to explain the incredible science behind these compounds safely and accurately.
So we leave you with a final thought to ponder. How does a molecule that unlocks ancient visions also hold the key to modern regenerative medicine? The Harmala alkaloids are the ultimate biological paradox. On their own, they're just mild, nausea-inducing stagehands. But put them in combination, and they act as a powerful pharmacological lever, turning inert DMT into a profound experience, and potentially helping us regrow the very cells we lose to modern diseases. They are a testament to the incredible, dangerous, and healing versatility of nature. Thanks for joining us for this explainer, and keep questioning the world around you.